Well, it is 7:05, so I think we'll give us a start here. I'd like to welcome those of you who are joining us. We're kind of from all over the place. We have a couple different states represented, which is nice. But I'd like to welcome you to our webinar on Morels. Bill Bakaitis is going to be our presenter this evening. And as I mentioned, this is a continuing webinar series on mushrooms. So the two others were the introduction to mycology, which is the study of mushrooms. And then we did the ecological importance of fungi as well as this one on morels. So we're going very specific this time onto everyone's favorite type of mushroom, the morel. I think there's at least one state anyway where that is the state mushroom. So Morel is the state mushroom of Minnesota, if you're wondering. So my name is Lauren Bohr. I'm one of the education coordinators. I'm the education outreach coordinator at Mohonk Preserve. And I'm going to be facilitating this webinar this evening. Feel free to use the chat if you have questions. Bill will give everyone a chance to, we'll have points during the presentation where we can ask questions, and definitely at the end, we will leave time for some questions that you may have for him. So before I introduce Bill, just a couple of reminders. We're all muted, which is great. We're pros at this now, so we're all muted. If you could stay muted, that way we don't have any distractions during our presentation. It's also helpful if you turn off your camera during the presentation, just for less distractions. I know that Bill is going to be sharing his screen with us as well. So we'll do that. And again, if you have questions, we can pop them in the chat. This presentation is being recorded. So it will be available for you later on to watch it again, relive it all. If your internet goes out or something, it will be available. I'll share the links with you once they are up and posted. And for anyone who couldn't make it this evening, they can watch it afterwards too. So it is now with my great pleasure to introduce our presenter this evening. His name is Bill Bakaitis. Bill taught at Dutchess Community College for 38 years before retiring in 2006. During his teaching career, he was granted sabbaticals to study graduate level mycology at SUNY New Paltz and at the New York State Museum in Albany. And that's where he worked with John Haynes, the New York State mycologist. He's a popular speaker, so we have had a lot of interest in his presentations along the way. And he's given a lot of educational programs in mycology at the Institute of Ecosystem Studies, and that's over in Millbrook, New York, the Culinary Institute of America over in Hyde Park, and Hudsonia at Bard College, as well as many other places throughout the Northeast. In 1983, he founded the Mid-Hudson Mycological Association. And since 1984, he's worked with poison control groups throughout the Northeast. So you can imagine maybe there might be some misidentification of some mushrooms that has happened. His articles have been published in the New York State Conservationist, Adirondack Life, Mid-Hudson Magazine, Poughkeepsie Journal, Mushroom, the Journal of Wild Mushrooming, and he's a contributing editor of the Journal of Wild Mushrooming. So Bill has a lot of great knowledge. After all of that, he has a lot of great knowledge, and I know that morels are a really popular topic. It's everyone's favorite mushroom to go looking for. So without further ado, Bill is going to share with us his knowledge on morels. So I'm going to turn things over to you, Bill.
Okay, and I will share my screen with you all. What has happened here? Share. It's here. This is a new screen. I've never seen this before. Oh, maybe here's why. Here's why. There we go. All right. Slideshow from the beginning. There we go. And if I change my view over here so I can get all this out of there. I'll have to figure that out as we go along. Any hints on how to collapse this thing on the right here? Is me. I want to disappear.
You want to disappear?
Well, I don't. Yeah, I know who I am. If this Okay, okay, I'll figure, let's see here, maybe this is it. I don't know. It's not changing. Well,
You
okay,
can hide your thumbnail video.
can you see, what are you saying here? You're saying,
So you can have if you're seeing yourself and you don't want to see yourself, you can hide your thumbnail video. If you put your cursor over where your video is coming in, you'll be able
yeah,
to see
I see
there.
there's a box there, hide thumbnails. I do that.
Yep,
There it goes, it's gone now. Okay, good, okay, we're
there
good.
we go. Okay.
Right, and the thing at the bottom, I guess, will disappear too. I'll drag that down, or oh, we'll just leave that there. Okay. Oh, I keep clicking on things and before I want to do it. Okay. So I do want to go back to what we said here before, what Lauren was saying. This is the third program in a series I'm calling Introductory Mycology for the Curious Naturalist. And we're going to talk about morels here. I have 80 slides, so we want it done by 8 and leave room for questions. So it's going to cause me to talk fast, I think. I'm a teacher, as you know, and so if you've missed the first 2 programs, you can go to the Mohonk website and find them there and listen to them. So if I get into some technical material and point to that, that's where you can get those good answers to those questions there. This will be a program about morels, but I will be referring back to those conceptual ideas. Of the readings that you got, I rearranged these slightly. There are 2 of them here that are about collecting mushrooms. The one from Collecting Wild Mushrooms, part 1, the morels. I think you'll find that very informative and probably the best one you'll ever have read. At least that's what people tell me. The second one here is all about conservation, and particularly in the digital age when people can somehow grab a hold of your photos or whatnot and come in and really rape the area you've been looking at. The last 2 articles are about the problems of lead arsenate in apple orchards. So we'll talk about all of those right now. So on we go. This is a talk about morels, and that's what they look like. Nice collection of them. We're going to talk about some habitat and abandoned apple orchards. That's the issue here. First, we need to talk about the toxicity. There is a false morel, a group of them that contain deadly poisons. The poison is monomethylhydrazine. We know a lot about that because it's a rocket fuel, and the space agency and government a lot of people have looked at that. For mushrooms, it comes from a compound called gyromitrin, which is in certain mushrooms, and then it turns into monomethylhydrazine. And that boils at 87 degrees centigrade or 190 degrees Fahrenheit. Water boils at 212. So if you're cooking these mushrooms and you're standing over the stove and you are smelling the mushrooms, you can get hit by the toxin, but your guests or your clients will not. Gyromitra, Helvella, Sarcosphaera, and some Pizziza are mushrooms that will contain this toxin. And this is what false morels look like. They look somewhat like morels on the outside. They're pitted, they're an Escamite seed, so there are acai in there with spores in the acai. But when you cut the stem open, rather than being hollow, it's all chambered. And that's a real good way to tell the difference, easy to tell it in the field. Some people do eat these and some people get away with it. Some of them are more toxic than others. But as we'll see later in this program, it's really difficult to know what a species is. The other thing that we should mention is that raw morels are poison in and of themselves. So you need to cook them, and cooking them does not mean stir frying. It means they must be well cooked. You want the water in there to be boiling, the cream to be boiling. I'll give you a recipe at the end that shows you how to do all that. But one of the largest poison events in mushrooms we've had in the United States came from a wedding reception. I think it was wedding. There were a lot of people there, and there was morels in a salad, raw morels, and I think it was 128, 138 people ended up sick, and many in the hospital. On to look at morels. The first morels you're going to find in the season are these little things in the left here, the lower left. There are a subgroup of morels called Verpa, the genus Verpa, and there's a few species in there. But this is the one that's in the Northeast we find. It's Verpa conica, called the thimblecap. They appear before the leaves are out on the trees very early, and I've only found them rarely and almost always under apple. The Morchella semilibera or semilibra looks very similar, and many people have given me this saying it's Verpa bohemica, but everyone I've looked at turns out to be Morchella semilibra, or now we have a new name for it. Here's the differences here. Bohemica, which is found out in Chicago and in the Midwest, has, like the thimblecap, like Verpa conica, is attached only at the stem up there. It's with Morchella semilibera, the fruiting part, the placenta hangs over the edge like that. Furthermore, the semilibra, or punctipes we call it now, has a hollow stem, and Verpa bohemica is a stuffed stem. Verpa also has huge spores. There are two large spores in the aci, but with the semilibra, what you're going to find is eight of them, is usual in most other Ascomycetes. Bohemica is edible, but if people eat it for more than one day, often they get quite sick. They get staggered and sort of drunken and loss of coordination. Don't know why my screen is frozen. Oh, there we go. Okay. So this is a good look at Morchella semilibera. I misspelled this on an earlier program that I can't correct, so there should be an E in there. I've just got to call it semilibera rather than libera. So it's called punctipes now, and named after these little rough sandpaper-like ridges it has, little punctiles there. I find those this time of year, before the larger morels come out. And around here, I find them in rich lowlands, like a little bench next to a stream or a bog or a marsh. And butternut, that white walnut, I find to be a good indicator for them. The best part of them, of course, is the pitted head. Otherwise, they're a little tough and maybe even a little astringent. But that's, I'm trying to move something, I can't move it. Okay, that's how you go there. This is very interesting. Okay, black morels are going to be the big ones that come out here. And as you see, they're hollow inside, and you see all those little pits on the outside. And they can get quite dark, as you can see from this one right here. They get quite dark. They often appear very early in the season, here May 5th, and that was maybe the first morel I found of the year. But often they'll be alongside punctipes or maybe a week later. And the big crop of the blonde morels come in about another week or so. So in modern DNA taxonomy, there are what are called clades, and they use little restrictive enzymes to try to track the ancestry of whatever they're looking at. And there's lots and lots and lots of species of morels. But here in the Northeast, there's maybe 3 major groups of them, discounting Verpa. You've got the punctipes, and then you got the black morels, and you got the white morel and the blonde morels. And those tend to be 3 different groups. So in the black morels, the ridges are darker than the pits, and that will, in the field, that will distinguish the 2 of them. Most people don't like blacks as well as the blonde morels, but they'll collect them and eat them. They're just not as plentiful, and most people will tell you they don't taste as good. They're very difficult to see. Here's a close-up of one right here. I'm going to back the camera up so you'll see what kind of habitat it's in. But often it looks like maybe a shadow on the ground, or a shadow of a leaf. If I back up a little bit, you can see how it sort of blends into the background. And if I back up even further, how can you see them there? I mean, they're just really difficult to see. You sort of have to have a sixth sense about what you're looking at, about where they are. You can You'll get the hang of it after a while, but they're very difficult to see. They do appear over limestone strata. That's one good key that you can use. Actually, here's one which is growing out of limestone. You can see there's a slab of limestone here, right at the Hudson River in our territory. For those of you who are from afar, the Hudson River is where it is because the great tectonic plates of Africa and North America smacked up against one another. So limestone strata is often thrust up in the air like this. And you can often find that. And this one's growing right out of the edge of it. It's found an edge. Species, when they find an edge, do different things. So this is a good one here. Those of you who have read the articles, this is the Morel I was photographing when the boys with the cell phones came along and called all their friends to come and ravish the woods. There you go. And I guess this is as good a place as any to talk about the diversity of morels. When you look at them, they have many different shapes, and a technical name for that is called polymorphism, many shapes. So there's a great amount of, let's see if I can move a little bit better. Morels will show a great variation of form caused by both genetic and environmental factors. I was telling Lauren before we started that my Microsoft program just stopped today. I don't know what it is. So I'm going to talk about complexity here in a complex fashion. It's only going to take for 2 or 3 minutes or 2 or 3 slides. So after that, we'll return to just common talk. But this is how professionals will talk about this. Each morel may be, and I can't read that. May be composed of a variable mixture of mono, di, multi, or heterokaryotic hyphae, and the pairings of haploid nuclei can occur anywhere. Such colonies should be considered populations of nuclei or genes rather than as individual morels. This inherent genetic diversity can apparently be augmented by the incorporation of new genetic material on a seasonal basis into an already established colony. Now that's a mouthful, and this is one of the major papers written on Morel ecology in North America. It's called the General Technical Review 710. So what this suggests, though, is that you can get regionally endemic species. And what that means is you can find a species of morel in your neck of the woods that does not exist in any other neck of the woods. It's yours. It's regionally endemic, and lots of them, but it doesn't appear anywhere else in the world. And I'll show you why that is. If I can get this machine to work, and we'll go down why that is. No morels ever analyzed were found to be genetically identical, even if growing from the same cluster. There's a cluster of morels, and every one of those is genetically different. Now, within a few years, we're going to be able to get cell phones or a machine like a cell phone, which will be able to sequence your mushroom, and it'll cost a few $100s. And that's in the works right now. But what's that going to tell us once we can get a good bead on what this genetics of this particular mushroom is, if all of them tend to be different? Here's if I just harken back to one of the previous talks, the first talk, this is the typical life cycle of a fungus, okay? And in the earlier talk, we talked about basidiomycetes. This is an ascomycete, so it's even a little more rich than the basidiomycetes. There is a typical dikaryotic development, and dikaryotic development, for those of you who are new to this, means that you get one set of genes from your mom, one set from your dad, but they don't. They get together in the juices of the hyphae. It's called plasmogamy. The juices get together, but the carrion, the nuts, the genetic material do not join. There is a prolonged period of a dikaryotic mycelium, and it's only in the placenta, in this case, it would be the pits of the mushroom, where the aci are formed and where you get the spores then becoming the fully diploid number of 2n, and then they go on to meiosis and mitosis and single N, and that becomes one cycle. So that's a typical dikaryotic cycle. They can make a mushroom, a morel. Morels can also be made by simply the spores just from one parent and not from another parent. And so what you've got here is you can find morels being formed by a haploid development, a dikaryotic development, or a diploid development. Now, I'm going to get in trouble here, but I'm going to try to simplify this. In science, in philosophy of science and logic, there's a concept called pathetic fallacy. It doesn't mean what you think it means. It means trying to make things emotionally, things which are cognitively precise, to make them emotionally understandable. So I'm gonna get into trouble with this, but let me try this. It's almost as though like you've got homosexuals, bisexuals, and heterosexuals when you make morels. That's the kind of genetic composition they have. Now, that's of the fruiting body. You've also got morels that only make mold. They don't make a morel. And this is the exact same genes, but they don't look like morels at all. Here are two morels that are now cultivated in China. Okay, we don't have them around here. And this is the mold, and this is what it looks like in the laboratory. And this structure and this structure which makes spores are completely different from these structures, and yet they are the same species. So we call this the anamorph, or the imperfect stage. This is the teleomorph, or the perfect fruiting body, and together they're called the holomorph. Okay? So the last thing I want to say about this, a lot of people who think about mushrooms think mushrooms are the end all and be all of everything. I think that's particularly true of people who eat mushrooms that get you high. And then in that state, they say, oh my gosh, I've seen God, I've seen the world. That's what mushrooms are all about. Mushrooms ain't much different than other things in the world. There are a few things that are different, but the same thing going on in mushrooms is going on in other places. One of the things in mushrooms is we don't see what's happening underground. So then we make up things, we invent things, we project what we want, need, and expect to see there. If you go to the Cornell lab and look up their BirdLife magazine, you'll find lots and lots of articles there about genetics and about species of birds. One I want to point to by, I think it's Roberta Heisman, What's in a Name, Genome Mapping. There's a real difference when you look at the entire genome of an organism rather than looking at restrictive enzymes. So what they're finding out is the closer you look at the genetic data, the more you come closer to looking at the entire genome, the more difficult, not the less difficult, but the more difficult it becomes to determine species. And they will have, in that journal, you will see 8 species of birds which are genetically identical. Well, 99.97% identical genetically. And yet they know that they're not the same. They don't breed together. One species will breed only with itself. It has its own bird call. It makes its own kind of nest. It has its own habitat. It has nothing to do in life sciences with the other one. The genes are the same, but they act quite differently. So that's probably, I'll leave it at that point. It becomes a wonderful thing to have another program about, which we may do sometime in the future. I don't know. But now let's go back to the normal kind of field mycology that you and I and everyone else do. The Morel season starts early. You can see this woman here dressed in down, maybe a day like today, going out and looking for morels. And right there is a tiny black morel. That's the object of the search. So you can see, and how do you find that in that kind of welter of stuff that's out there? So when do you start looking? Well, forsythia. When the forsythia petals, the blossoms, start to fall on the ground, I find that to be a very reliable indicator that morels, black morels, are up. It's often said that when the oak leaf is in the mouse ear stage, when it's just starting to come out and looking like a mouse ear, that's when you're going to find morels coming out. If you get out, I found morels around here from the last part of March all the way through the middle of June. There's a lot of habitat in this area, and I'm quite sure now down near Philadelphia, you've got morels up down there now. I'm quite sure of that. But you will find a lot of April Fools. A good seasonal indicator around here is the apple bloom. When apple trees are in blossom right now, that's when the morels are going to start to come out. Mine right now in my yard are in that red tip form, so they'll be coming out soon. The other good indicator is lilacs. When you see lilacs blossoming, and I have a couple of them now just about to bud burst, then you're going to find morels. Essentially from May Day to Mother's Day, time to look for them. Now, there are 3 clades around here. You're going to find the punctipes, the black morels, and the blonde morels. And let's look at some blonde morels. A lot of different names are used. Esculenta means edible. That's the name you're going to find in most field guides. Esculentoides, oides means in the face of or looks like. Americana, because we're America. Ulmaria, because it grows under elm trees. Lots and lots of new names. 130 different species worldwide. They usually appear in upstate New York and Massachusetts, Connecticut by mid-month May. And here you see, and this is May 12th, they were up. So that's what they look like. I should probably, no, maybe I'll do it later. That knife is 3 inches long, closed. Okay, the basket is pretty big. I'll show you the basket later if we have a chance. Now with blonde morels, the ridges are lighter than the pits. So the fact that this morel looks dark, the ridges being lighter would segregate it, at least for field identification. And you'd call that a blonde morel. Doesn't look blonde yet, but it will mature that way. Young esculentas are going to be gray. Often they're the same gray as the peeling bark that's fallen from the elm trees. But that's them. But as the spores mature, they start to get orange, and they will throw a lot of spores. If you want to dry these morels in, let's say, your oven with a pilot or under a light bulb, be prepared. They're going to throw spores all over the place. The prolific sporulators. With both black and blonde morels, they're going to have a hollow interior. Okay. Even though this one here starts to look like a little convolution there, but it's going to be hollow essentially, and hollow inside. The blondes come out later in the year, so lots of insects get into them, and a lot of insect damage and insect, what do you call this? Insect shit, I guess you call that. But that you find a lot of that. Every now and then you'll find some with red stains. That's perfectly fine. It's perfectly natural, not a problem at all. Later in the year, as the insects come to love them, you're going to find insects living there. Sometimes a few ants, but often a whole ball of ants, like a whole ant colony with their eggs and the queen in there. You know, and what do you do with that when you pick it up and they're now crawling up your sleeves and everything else? Holy cow. You'll find salamanders, you're going to find doodle bugs, lots of things go and live inside of there. For blondes, a very good seasonal indicator is asparagus. So when the asparagus in my garden is in this stage right here, that's when the blonde morels are doing well. Trout and fiddleheads, they say the same thing about trout, that when the oak leaf is the mouse ear stage, trout will rise to a dry fly.
I'm not telling you this to like be like, oh no, it's like not true. No, there are people taking this seriously.
Hello, what's he saying?
Oh,
I'm sorry, what's happening?
Go around trying to write people, make threats like that.
if you could mute yourself.
So I'll tell you about this shot here. I was fishing in a local river for trout. These are put and take trout, stock trout. And I'd seen a place where a beaver had gone up the hill. It was a nice beaver slide. And I was curious. I just walked up there, and sure enough, there was an elm tree that apparently he had killed. And at that point, there were a lot of morels there. So I said, well, this could be a good shot. So I went back to the car, I got my camera. This was when I was using 35 millimeter, I think. So I set up the camera, put my creel there, put up a lot of morels, found some fiddleheads, went back, caught this trout and brought it up right away, snapped this shot and brought it back and it swam away. But this gives you a good indication of what's happening in springtime, a lot of things all at once. One of the best indicators for morels are recently dead elm trees. And here you find, here's a dead elm tree here, probably no morels under that. But this one here, there would be morels there. This is me, and these are a couple of chefs in the culinary. I did their mycology classes for them for about 30 years. So we would go out at least twice a year, and every year with a new group of chefs and students. But this is a good morel habitat right there, what you're looking at. This is a recently dead elm tree. A few years ago, I saw this between a development and a shopping center, and I stopped and looked, and there were a good pack of morels under that tree. The next year, there was only 2. That's the way they go. They come and go very quickly. A good indicator of morels is when you find the cracked bark of infected elms. So there's a progression here. There are going to be galleries here. The chestnut elm disease, the beetles carry that fungus in, and the larvae make little galleries, and the larvae actually eat the mycelium of the mushroom and the cambium layer. And what they leave is a lot of heavy, big sugar molecules which clog the cambium layer, and the elm tree dies. And often the bark's cracked because there's another fungus there, Flammulina velutipes, which just pushes the bark off the tree. So that's a really good sign. You'll be able to find those there quite, well, not every time. That's a good sign. You probably have to look at about 50 elms before you find morels. And this again is another group of culinary students out. This is a good look at good morel habitat, a big, nice elm tree. If you have trouble finding elms, they often make these long arms that go out. And when you find a morel, you find one here maybe and one here. They're growing right along the root zone there, called the rhizosphere. And if you follow that out, you will find morels often, they'll grow right along that root zone. Maybe I can talk about that root zone a little bit later when we talk about pH. So here's a good picture of good elm habitat. And we can talk a little bit about some of the morel habitat here. Morels are sometimes saprophytic, sometimes mycorrhizal, different parts of their life cycle. They're called functional mycorrhizal. They're not truly mycorrhizal, but they work as if they are. From one of the earlier talks, you'll know that I mentioned that there's often a mycorrhiza often can be described as mutual parasitism, because the way that one organism eats its way into the other organism and feeds the first one. Then there's a new kind of relationship which we're learning more about. It's called endophytic. Endophytic means that fungi will grow inside of the cells of a grass, of a shrub, of a flower, of a plant, of a tree. And what we're now finding is that there can be endophytic symbiosis, which means that the hyphae of a fungus, which is growing inside of the leaf of a plant, and almost all leaves apparently have this. And you can Google this and see pictures of these. But it passes the fungus on to the new plant through the seed. And this is something which, what's her name? Dr. Jean, those of you who are watching these Zooms now, Dr. Jean Lodge, talking about a guild mushroom, hygrosabe, was talking about that just a month or two ago. The brand new concept to me that fungi can be passed on to through the seeds, but there you go. So what you find here is that these morels are infecting one tree, and I say this one here, and then as that tree dies, They go on to infect another tree, maybe this one here. And then as that one dies, they move on to infect a third one. And there are at least two very easy ways we can talk about this. One of them is that the beetles that are here will carry it to another tree. The other one is that there are root grafts underground. An infected tree will graft with the root of another healthy tree. And when that happens, the infection can go from tree to tree. So when you are out looking for morels, when you find a dead elm tree, you want to look around that area and see what other trees are there. And chances are that if you find a tree that's productive, that production will spread to another tree in that general area over a few years. In this tree, it'll last there. The first year, you'll get a prolific fruiting. The second year, you might get a fruiting of maybe a dozen. The third year, maybe 2 or 3. And after that, it's pretty much the end of it. And actually, that's exactly what Chinese are finding now when they're cultivating their mushrooms in forests. They find that they have to keep moving the forest production because it's only good for about 3 years. And those are all saprophytic morel species that they're using in China. If you can't find elms, another thing to look for are ash trees. And here you find a nice ash tree. Again, these are some chefs and students at the Culinary Institute. And we had been under this ash tree here, we found morels for 6 or 8 years in a row. And then one spring we came and the area had been raked through, I mean, really raked, everything had been raked out. And from that point on, there have been no morels found there. So, you know, it's sad to say that people will often go there and take the ball before you in that way and destroy the habitat. As you know, most of you know, the emerald ash borer is destroying most of the ash trees in the Northeast right now. So here is a healthy ash on the left, that really nice gray bark with that sharp diamond-shaped fissures. And this is one which has been infected by the emerald ash borer. A few other signs too, but when it may be worth looking at these when you see these trees, and they're all over the place now. Every now and then you'll find them under ash. I find them there, but not nearly as productive as elm. Apple orchards we're going to talk about in a bit. Another good indicator is garlic mustard, and you're going to find garlic mustard. It's an endemic invasive species now. They just takes over very easily, and it provides a lot of good habitat, I guess. Certainly it's a good indicator, seasonal indicator. Ladies' bedstraw is a good indicator. There are 27 morels growing right in this one image right here in this bed of bedstraw. And sweet soils is said to be a good indicator. And around here, that means limestone. Maybe I should just say a few things about sweet soil. It's really not sweet that they're growing. They fruit best at about 6.5 to 7. Okay, 7 is neutral. Below that is very alkaline. Above that, I should say. Numbers below that are very acidic. But with most rain is acid, and a lot of life processes are acid as well. Think of lactic acid or fatty acids. Morels grow very well right in this area right here, just where milk is in this. But what happens to the soil is that there's a lot of rain that comes, particularly acid rain, or just as the soil in a forest ages, it becomes more acid. There's a potassium hydrogen ion exchange in the root zone of trees, and the soil becomes more acid. So what you want to do is you want to bring that acidity down, and there are a number of things that will bring it down to this helpful level right here. Certainly limestone is. If you find limestone in an area, you're good chance of finding morels there. So 3 things I can think of that'll bring the acidity down, the soil down. One is limestone, and I've found lots of morels on the edges of fields that are well lined, like cornfields or hay fields that are well lined. That's a good place to look. Another place to look are railroad beds, particularly old railroad beds where they had old steam engines that burn coal or even wood, because as the wood ash comes out, that also alters the pH of the soil around there. And you find that. And the third, burn sites out west are very good places. I've looked at many burn sites here in the east and never found anything that produced morels, but out west they do. But there is, when I talk about the root zones, I don't have the literature with me. I brought it all over to the Mohawk before the pandemic, and it can't get in anymore. So it's all over there. But these are studies I'm referring to that were conducted by Institute of Ecosystem Study. You know, Gene Likens was there and did a lot of work on acid rain. One of the things they find is that in the root zone of a tree, as the tree matures, it becomes more acid. But when the tree dies, there's a sudden change right in where the roots are. And when you look at apple trees and you look at probably elm trees, that's where you find the morels coming up. So that seems to be one process by which the soil becomes more alkaline in that area. And then the third thing are clay soils. Clay is normally very alkaline, and you know, just pucker your hands up to be nothing if you're working with it. But when acid rain comes and meets that, often the cations are released, and you find that often soil that is acid suddenly becomes alkaline. So in clay-based soils, you'll often find morels there as well. Sorry, I'm talking so fast. I'm trying to get through this in 50 minutes here, 45 minutes. Acid rain will cause limestone to flow. You put a drop of it on hydrochloric acid, it'll fizz. You see lots of limestone around in colonial furnaces. Bridge construction, there's an old limestone bridge on an old farm road. If you get some geological quadrangles and you find where the calcium soil is limestone soil, that's where you want to look for morels. You don't want to look in harder other rocks. Later in the year, the morels turn into big blondes. These are often called crassipes, but they are just as Esculent has grown larger. There's another very small morel, which appears early into June. They're often called deliciosa or tulipfera because they grow under tulip trees. Now I think they're called virginiana with DNA. I often find them under upland skunk cabbage, not the skunk cabbage that grows in bogs, but in dry soils. So that's often a good indicator for me. In apple orchards, they can be very productive, but they're not without danger. And here's how you find your orchards. If you get some topo maps, they'll show you orchards. And orchards are marked with this kind of indicator you see right there. And if you just look then through the gazetteers, you're going to find where there's going to be nice descriptions of where orchards are. Along the Hudson River, there's a lot of orchards. There's a problem there, though, that a lot of them are posted. They're often still in production, and they're often on private land. There's a lot of gentrification going on through this area, and the new people who come in own the property, and they don't want anyone else there. So it's very different than it was, say, 30 or 40 years ago. The other thing to keep in mind is that May is both morel season and turkey hunting season. So there'll often be turkey hunters in the woods around here. This is a road. I know the morels at the end of that road. I've been there. New owner posted it, can't get in. And this elm tree here, boy, I would like to see that. I found them under this elm tree, and there's an apple orchard back there, which had them, but you can't get there anymore. As far as COVID goes, last year, there was a good line of demarcation right here at the Columbia, Dutchess, Ulster County border. Down here was high rates of infection. Above that were not. Using the same map because the new map won't reproduce for me. What we find is that there's essentially the same area. The infection rates, the protocol for measuring infection has changed from last year to this year. So these are not exactly comparable, but you do see that in this area, they're higher. Statewide, it's about 2%, up 2% up in this area. But this weekend in the New York Times carried an article on the extreme risk of this area right through here of Orange County and adjacent Pennsylvania, all the way into the Poconos and down into New Jersey. An extreme risk, they call that. The other areas are very high risk. So as you go out, you take that in consideration. You often find orchards just by driving down country roads looking for hillsides covered with bloom. They can be productive Here's just one walking between one row of apple trees, May 11th, 2008. And that's about almost a half bushel, that Creole. But by 2016, the orchard was impenetrable. Vines, snowstorms toppled the trees. The orchard's now gone. This is how you get into it. You know, there are a dozen or so morels in this picture. Here's 1, 2, 3, 4, 5, 6, 7, 8. So you see how they are there. If you're going to go in there, you want knee-high boots, leather gloves, clippers. You will need to cut yourself in and out of these orchards. Here, for example, is someone going into an orchard on their hands and knees, and you get cuts and scrapes and lots of other things. This is a deer tick, and this is area central for Lyme disease, right around here. So there's one of the dangers you've got. Here's another, it's poison ivy. Poison ivy is prolific in these old orchards. Would you eat these morels? Having worked with poison control, I can tell you we get cases where people have eaten mushrooms collected in poison ivy and they break out in a rash from stem to stern, top to bottom. Eventually the orchards are going to disappear and they're going to be swallowed up by a reclaimed forest. And now this brings a new danger, because you won't know if that area had been treated when it was an orchard with lead arsenate. These are my handwritten notes showing a great deal of variation in arsenic and lead in orchards. There's a number of orchards here. There's a great deal of variation within orchards and between orchards. So and what we're talking about here are not things, these are elements. Arsenic and lead are elements. They don't break down. They're going to remain there even in the succession forest. They'll usually be in the top layers of duff, top 8 inches or so. They're going to be toxic forever, and they seem to adhere to morels. Okay, so in these areas you can go there and you can find morels, but you won't know if they have been infected with lead. Part of the reason for that is that morels grow from a sclerotia. The sclerotia was formed last year, and it's underground. And if it's not a full sclerotia, it'll be what's called a muff, a more loose organization. And there's a lot of dirt there, and that dirt tends to adhere to the pits and surface of the morels. This is Dr. Ginstein, who came and collected some morels in this area a few years ago. And I took him to places where I knew they had used lead arsenate in the orchards, and we collected morels and he brought them back. And this is what he found under his electron microscope. This is assumed to be the articles of lead that are in the pits, well in the pits of the morel. There was so much lead there that the next year, a student and professor from Tisch at NYU wanted to come up and collect morels to use in their art projects. They wanted to use the morels to react with their glazes to make special kinds of pigments. So collecting in an open forest is a lot easier and safer. Whenever I'm out in an open forest, I do a lot of walking and a lot of driving around, carry a big basket, another bag inside, because you never know when you'll hit the jackpot. So if you're lucky enough to find a lot, you're going to clean them well, and you want to check inside for centipedes and salamanders and whatnot. Here's a good simple dinner at this variation on Morels Rosenthal. Put some butter in a pan, garlic and shallots, a few caraway seeds, and if you have a bell pepper, a few pieces of bell pepper. The caraway and the bell pepper really accentuate the umami of the culinary flavors of the morel. Once the shallots start to get translucent, put your morels in, split them in half, make sure there are no bugs, put them in face down like this first, cover them with a lid, and they will start to cook in their own juices. And you cook them that way for 5 minutes or so, maybe 10 minutes. Then you make a nice cream sauce. Chicken stock makes a good cream sauce. Arrowroot flour makes one of the best. A little bit of tamari, heavy cream, finish with port or Madeira or sherry, fresh pat of butter, seasoned to taste. There it goes. And you can serve it over pasta, garnish with ramps. And I like these ramps to be grilled and charred. And this is one of the best. If you have a lot left over, you can make some Dixels and dry the extras for the winter. And I dry a lot of them off in my greenhouse, just like that. But there you go. Last May 18th and 19th, the season was drawing to an end. And this is what I found on those 2 days. Some ramps were still around in the highlands. There was, these were the Morels I found. And Stropharia was growing in my garden, and this with the asparagus. So I've talked fast, quick. Hope I've left room for questions for you. Good luck, and let's do that. I will stop sharing my screen here if I can do that, and we can take your questions.
Yeah, we can open it up for questions. I don't see any in the chat right now.
I think we should have a round of applause.
Well, thank you.
That was tremendous. That was tremendous.
Thank you.
I took a lot of screenshots and I learned a lot.
From your car?
From my car. I'm at my son's soccer game. It's starting in 7:15.
You're a good dad. You know, I have a party for a 94-year-old at 8 o'clock, but I'm willing to go a little longer because she'll be around for a bit.
Okay. All right.
Zoom. Isn't it amazing what we do in Zoom? Yeah. Who are you? Where are you?
My name's Will. I'm in the middle west of America, specifically Schaumburg, Illinois at the moment.
Uh-huh.
Where Married with Children was set, for people who don't know, the television program. But anyways, I've been out once. I got ramps, and then as someone called it, the carp of the forest, the pheasant back.
Oh yeah. It's Polyporus squamosus. Yeah. A lot of people like that. I know some people who like that better than Morels. You do too.
Early season, very small, fresh.
Yeah.
And just chopped up in a pan sauce after the ribeye was in it. It wasn't bad. It wasn't bad.
Yeah. Okay. Well, let's see who else has questions.
Yeah, I saw a hand raised. I think it was PJ's Scott 1 had a hand raised. If you have a question.
PJ. Scott 1.
Oh, maybe the question was answered. There is a question in the chat, and the question is Morel picking allowed on Mohonk Preserve? No, it is not allowed on the preserve, just because we are a land trust, we are a land preserve, and we do not encourage people to go off trail, create social trails and things like that, and then harvesting anything from our land. But you're welcome to look on the side of the trails for some, just please. I see Anthony. Anthony, you have your hand raised. You can go ahead and ask a question.
Good evening, everyone. So Bill, I'm down in Rockland County. I'm a former chef.
Former chef.
Yeah. So that, by the way, nice job on that mushroom dish with the sherry deglazing. I like that. You did good. So, so real quick, do you do any tours that I could join you guys up there in Orange County? Is there anything that you have paid tours or volunteer tours that you could help me with? Because I did go out and purchase the Audubon Society Field Guide to North America mushrooms, but I do find it difficult sometimes trying to pick the right mushroom and figure this all out.
Yeah, I don't think you'll have trouble with Morels. There are mycological associations which have, well, in non-COVID times have walks. And actually in COVID times, they're having walks. I don't know how you maintain distance and whatnot, but most people are getting vaccinated now. You know, most of the people I know are now vaccinated. I think, you know,
I am.
Yeah, most people can now walk. I think it's now any adult can walk into places, and I get my cell phone pings all day where this place is open, that place is open. Yeah. So
right. I got lucky once up in the Neversink trout fishing. I found a troops of chanterelles, and I stopped fishing, and my brother he couldn't figure out why I didn't want to fish anymore once I found chanterelles. So I just couldn't leave the woods after that point.
Do you carry bear spray with you in the Neversink?
No.
I've been told that's the thing to do there.
Yeah, I hear that too.
Yeah. Well, but if you look at NAMA, N-A-M-A, North American Mycological Association, if you Google those, you will find a list of mycological associations throughout the area, and you can join those. And
could you say that one more time? And what was that? The acronym?
North America Mycological Association.
Got it.
NAMA.
Yep, got it.
There's a trucking firm and a couple other things too, but if you put in NAMA Mycology, you'll get it. It's a great website, great resource.
Okay,
Yeah.
I'll look it up. Thank
Chef,
you for your time.
where are you cooking? Where do you cook, sir?
I'm retired as a chef, so I was down in
What?
New York. Yeah, I was down in Manhattan
What
for
restaurant?
Le
What restaurant?
Caravel, Le Cote Basse. So I got graduated culinary up at Hyde Park and went down there in the mid '80s.
Did you take
And
courses with
nice.
me there? Did we go on walks together at the culinary? I was there for 30 or 40 years.
85, so I don't remember you.
Oh, that might have been right before.
Mhm.
It was Jay Stein started. Jay and I started that. Yeah. Yep. There you go.
Cool.
Love the outdoors though. I'm always trout fishing, hunting, everything. So
Who else?
mushroom hunting just adds to the reason to be outside.
Yeah, I don't want to cut you
Something to
short.
shoot.
Yep.
Well, we only got a few minutes, and there are probably other people who have questions too.
Agree.
Yeah.
Yeah, we can open up to, it is right at 8 o'clock, but if anyone would like to have their burning question answered, I'm happy to do. Yeah.
Anthony Simi has a hand up.
Oh, no, I think he already had his question. I think his hand still up. There we go. Yeah. I know some of you may need to head out, but thank you so much for joining us. This was a great presentation. I learned an awful lot about morels and where to find them, and different ways of cooking them too. So if you happen to join in a little late, that is okay. This has been recorded. So I'll be sending out a link with the recording probably next week when it all gets edited and all that fun stuff ready to be posted. So you'll be getting that. Check your email for that as well. And of course, check your email and our website for future programming too. Right?
Thank you for your time. Appreciate it.
Thanks for joining us this evening.
Nice questions. Thank you. Right.
Yeah, you just have a lot of thank yous. Wonderful presentation. Enjoyable and inspiring. So I think you inspired a lot of people to get outdoors and look for some morels, and now they know where to find them. Hopefully avoid the
yeah. Okay. I'll hang around here as long as there are people there in case someone else.
I see people checking out. That's okay.
Will still has something he wants to say. Will, I can't hear you.
Yeah, Will, you're muted.
I muted myself so I wouldn't keep butting in because I know it's being a hog. But ramps, right? So I went out last week and I found ramps. They seem really fresh and new. I did not find a morel. I'm pretty sure the morels are there that I didn't see them.
Yeah, it could be.
But,
yeah.
But if ramps are up, what does that indicate in regards to the morel?
And Tommy, around here, ramps come up about 2 weeks before morels.
Okay, good to know.
Yeah, they come up 2 weeks before morels. Morels generally, I think the soil temperature should be around 53 degrees before they start to come up. Ramps could come up, I wouldn't say through the snow, but right after the snow goes, once they're up quickly.
Got as a bulb.
One of the things about ramps I should mention, unless you have a really huge supply, it takes 100 years for a ramp patch to become sustainable. So they can be ravaged very quickly, very quickly. There's an area I've been collecting in where I'm the only person that's ever been gone there for 40 years. But this year I found someone came in with a tractor. This is a forest. They came in with a tractor, and I would say the area I was collecting ramps was probably about 3 or 4 acres. Covered a whole hillside where there was a settlement there years ago. Now someone came in on an old road with a 4-wheel drive vehicle and plowed up for whatever, I do not bring things to restaurants, or I don't bring them to farmers markets. My feeling is that if you really want these wild things, you should educate yourself, learn how they are, where they are. Half the fun is in finding them. And I think I'm morally opposed to someone being able to buy into something that because they have money. You know, I know I'm speaking here, you know, the people who are chefs are not going to like this, but you know, just because you have money doesn't mean that, well, you know, you don't go out and shoot wild rabbits and and squirrels and things and then sell them in your local restaurant. I mean, those are farmed, you know, you farm rabbits and you know, you use plump New Zealands in your restaurant, you know. I think Morels, you know, you know, out west, they get into wars. They shoot people if they who are taking the morel,
Poachers.
and then they set the forests on fire just to have more morels next year.
Well,
I know. Oh, yeah.
yeah, I did see, and this will be the very final one. I saw that Helen had her hand raised, and maybe maybe your question was answered or not. But Helen, you can go ahead. Oh, or maybe Helen's question was answered.
There was someone who raised a comment, which I didn't hear during the talk. It sounded like it was a disparaging comment. I'd love to try to handle it, though.
Yeah.
The comment that was made during the talk?
Yeah.
I thought that was someone with an unmuted microphone having a separate.
Oh, I see.
Am I right? If I'm not mistaken?
Okay.
Someone was talking
like
in the background.
Yeah.
A side conversation. No.
Gotcha. Okay.
Yeah.
No one was criticizing the mycology.
That's right. No one was. Well, all right. I think we will call it an evening. Thank you, everybody, for joining us. I really appreciate it. And keep your eyes out for future programming.
Yeah.
Okay.
Have a good evening, everybody.
Bill.
Yeah, maybe we'll see you over the Mohonk when everything is open again.
That's right. Yes.
Sure.